The information that our society currently has available on the safety and efficacy of vaccines is not settled science. Actually, very little of science is settled when framed in the totality of the realm of science. Science is a forever developing field that oftentimes has to go back and repudiate the previous findings based on new data. Therefore, it is imprudent to require vaccinations on the premise that it is for the protection of the public health, when these same vaccinations may indeed be harming the public health. I will give an example. The example is technical and detailed, but I believe necessary to understand the complexity of the issue in which we are dealing. Mercury is a neurotoxic metal. People are exposed to mercury from a variety of relatively common environmental sources, from dental amalgam, tooth fillings, to fish consumption. Mercury can also be a component of some vaccines, notably those in multi-dose preparations. Gene-mercury interaction studies have identified some common genetic variants that worsen the adverse effects of mercury found in polymorphisms common in individuals. A polymorphism is the presence of two or more variant forms of a specific DNA sequence. Two examples of gene polymorphisms that affect mercury neurotoxicity are genetic variants in the copropore fibrinogen oxidase, which we shorten to say CPOX, and the catechol O-methyltransferase, COMT. About 28% of the population have a CPOX gene that encodes a variant form of the CPOX called CPOX4, which substantially reduces heme synthesis. Heme synthesis is part of a red blood cell. The altered heme synthesis affects the individual's ability to tolerate mercury exposure, even low-level mercury exposure. The effects of low-level mercury exposure on those with this polymorphism are wide-ranging, including deficits in attention, visual spatial acuity, executive function, learning, memory, and motor function issues. About 1/2 of the population have the polymorphism COMT RX4680, which independently predisposes them to many of the same disorders of attention, learning, and memory as does mercury exposure. Accordingly, people genotyped with the COMT RS4680 have been observed to be especially susceptible to the neurobehavioral effects of mercury. Thus, while either the COMT RS4680 or mercury has a limited impact on a person's neurobehavioral performance, together, this polymorphism plus the mercury exposure results in greater deficits in tests of attention, learning, and memory, and visual spatial acuity compared to the mercury-exposed persons that are genotyped as the COMT WT. WT stands for wild type, and that's the non-variant form of the gene. It's the one that almost everyone has, except for the ones that have the polymorphism. To understand the detrimental effects to the population of these two polymorphisms, we have to make a calculation. You take the probability of event A. Event A is the percent of people with the CPOX4 gene variant. That's 28% of our population. Then you take the probability of event B, which is the percent of people with the COMT RX4680 gene variant, which is 50% of people have that variant. Because they are independent events, that one event is not dependent upon the other, they occur independently. To find the probability of having both those variants, you have to take the intersection of the probabilities. So you multiply them is what you do to get the intersection. So you would take the probability of A times the probability of B, which is 0.28 times 0.50 equals 0.14, or 14%. This means that there's a probability of 14% of our population that can carry both gene variants, exposing them to the risk of low-level mercury intolerance. That could result and greater deficits in tests of attention, visual spatial acuity, learning, memory, and this all is in a gene dose response manner. Now, gene dose is a term that refers to the number of copies of a very particular gene present in a genome. And different individuals can have different number of copies of the CPOX4 and the COMP-RS4680 gene variants. The 14% of our population that could carry the polymorphisms that predispose them to low-level mercury intolerance, resulting in neurobehavioral dysfunction, is only one. It's only one example in many that are science. And it shows us that science is not settled when it comes to vaccinations. There are numerous examples that I could bring to the committee to further elucidate this point, that we, as a scientific community, don't have all knowledge when it comes to vaccinations. My intention in bringing an example of this detail to the committee's attention is that it is imprudent for employers who may not have taken the time to study the full impact of their requirement for vaccination to have the power to require a medical procedure for individuals. The choice of whether a medical intervention should or should not be done must remain with each individual, not an employer. If an employer does decide to require an employee to receive an immunization as a condition of employment, a waiver must be available for each individual to independently make the determination if they will or will not take the vaccine. Mr. Chairman, I have testified to this committee as a biochemist, a nutritionist, and Master of Public Health candidate at the George Washington University in Washington, D.C. However, you and the committee should be made aware that as a representative of the people of the Wisconsin 68th District Assembly, I've added my name as a co-sponsor to AB 612 as of November 15th, 2023.